Hello and welcome to this episode of Science with Verity. Today we are going to be looking at coordination, which is how the different systems within the body work together. There are two main types of coordination in mammals. The nervous system, which is formed of neurons that pass electrical impulses along the length and target cells by secreting neurotransmitters. This results in rapid communication with short-lived responses that are localized. The hormonal system produces chemicals to target cells, and this is a slower, longer-lived, and more specific type of communication. Chemical mediators are transmitters at a cellular level. Chemicals which are released by infected or injured cells to affect others in their vicinity, causing swelling and a rise in temperature. Examples of these are histamine, which causes small arteries and capillaries to dilate to cause swelling and redness, and prostaglandins, which also cause the opening of arteries and capillaries, and so numb pain sensation. Plant growth factors are the equivalent of plant hormones, which cause responses to a variety of stimulus such as light, gravity, and water. The plant growth factors influence the growth of a plant and are released throughout the plant by cells, and can even affect the tissue that they are released from, rather than a target which is a distance away. IAA, or indole acetic acid, is one of these plant growth hormones and causes plants to elongate as a directional response to a light source. Young shoots bend towards the light because the light is used in photosynthesis. When the light source is aimed at the shoot, cells at the tip produce IAA, which is transported down the shoot and transported to all sides. The light causes a movement of IA from the light to the dark side of the plant, so a greater concentration of IAA builds up. When this happens, the cells elongate, growing faster so the shoot bends towards the light. A neuron is a specialized cell that is adapted to rapidly carry electrical impulses around the body. It is made up of various structures which enable it to function. It has a cell body containing a nucleus and rough endoplasmic reticulum, which are associated with the production of neurotransmitters. Dendrons, which are extensions of the cell body and subdivided into dendrites, which carry nerve impulse towards the cell body. An axon, which is the long fiber running the length of the cell. Schwann cells, which surround the axon, aiding with protection and electrical insulation. They play a part in phagocytosis and nerve regeneration. They are layered many times so the axon is wrapped in layers of membrane. Some neurons have a myelin sheath, which covers the axon. This is formed from Schwann cells. Neurons with this build-up are known as myelinated neurons, and those without are known as unmyelinated neurons. The nodes of Ranvier are gaps between Schwann cells that occur when there is no myelin sheath. There are three types of neuron, classified by their function. Sensory neurons carry impulses from the receptor to the intermediate neuron. Motor neurons carry the impulses from the intermediate neuron to the effector. And intermediate neurons are those that carry impulses between the other two types of neuron. A nerve impulse is transferred along the axon by a reversal of the potential difference either side of the membrane, causing a resting potential and an action potential. The resting potential is generated by a process using sodium-potassium pumps. Sodium ions are actively transported out of the axon, and potassium ions are transported into the axon. This sets up a chemical gradient, and there are more sodium ions being pumped out than potassium ions. Diffusion occurs, and the potassium is encouraged as potassium channels open, whereas the sodium channels remain closed, preventing their diffusion. The outside of the axon becomes more positive because of the influx of potassium ions into the axon. The potassium ions are then attracted to the negative state inside the ion, and repelled by the positive outside, making them remain inside the axon. Action potential is the result of a stimulus being received by a receptor, and the energy causing a reversal of the charges on the axon membrane. This action potential causes the membrane to become depolarized. The channels change shape and are now called voltage-gated channels. The energy of the stimulus causing some sodium gates to open. This causes an influx of sodium ions, and then this triggers a reversal in charge as they are positively charged themselves. This influx closes the voltage channels and the potassium voltage gates now open. With the sum of the channels open, the electrical gradient is reversed, causing more potassium channels to open. This causes repolarization of the axon, as more potassium ions diffuse from it. The outward diffusion causes a brief hyperpolarization, and the voltage-gated channels shut, preventing further diffusion. Resting potential is re-established as the sodium-potassium pumps actively transport the ions once again. The action potential travels along the neuron in one direction, and the size of it is the same all the way along. The stimulation of one region of the action potential is caused by the previous region's depolarization, almost like a Mexican wave. For an unmyelinated neuron, the potential is passed the whole way along. The analogy works like this. The resting potential of the axon is polarized, and this is like the whole stadium being seated. The stimulus causes an influx of sodium ions, and the membrane becomes depolarized, rather like the first line of people standing to wave their arms. The second line of people is stimulated by seeing the first line, rather like the influx of ions causing the gated channels to open further down the axon. The second line prompts the third line, whereas the first line sits down, much like the repolarization of the first region of the axon. Action potential traveling along an axon with a myelinated sheath is like a Mexican wave, but in larger sections, allowing for a more rapid transmission of the action potential. The myelin sheath acts as an insulator, so it prevents the formation of the action potential by blocking the reversal of charges. The nodes of Ranvier are the places where the action potential forms. The potential skips from node to node. This is what allows for the rapid transmission. The impulse is transmitted at speed because of several reasons. The myelin sheath acts as an insulator, so the jump is larger than in an unmyelinated sheath. The diameter of the axon aids the speed due to the lack of leakage of ions, making it easier to control the potentials. The temperature means that enzymes which work the sodium-potassium pumps are able to work in optimum conditions. So the higher the temperature, the faster the impulse. That is, until the temperature is too high and the enzymes denature. The refractory period means that the action potential moves in one way and separates them, so only a limited number can pass along the axon at one time. Synapses are the junction between neurons and effectors, making them the gaps at which the action potential is passed from neuron to neuron. Impulses are sent via neurotransmitters across the synaptic cleft, transported from vesicles in the synaptic knob, which is the globular end of the presynaptic neuron. Synapses are like junctions in that they allow an impulse along one neuron to be sent to a number of different neurons, allowing for simultaneous responses as well as a combination of impulses from different receptors, allowing for interaction between them to produce a single response. The features of the synapse include its unidirectionality, in which impulses pass one way along the neuron, meaning that the impulse is only generated at the postsynaptic neuron, and the summations, where a sufficient amount of neurotransmitter is built up to exceed the threshold so that a new action potential can be generated. Temporal summation is the repeated release of impulses over a short period of time so that enough transmitter is released. And spatial summation is when a large amount of transmitter is released at the same time from different presynaptic neurons. Acetylcholine is a neurotransmitter that is released by a specific type of synapse known as a cholinergic synapse. These are common in vertebrates found at neuromuscular junctions. The arrival of the action potential releases calcium ions, and these enter the synaptic knob via calcium channels. The ionic influx causes vesicles to bind with presynaptic membrane, causing the release of the neurotransmitter acetylcholine. The transmitter binds with receptors on the postsynaptic membrane after diffusing across the synaptic cleft, opening the sodium channels which generate the action potential. An enzyme breaks down the acetylcholine into ethanoic acid and choline, which diffuse back across the synaptic cleft to the presynaptic neuron. This breakdown prevents more action potentials being generated. ATP is then used to recombine the elements of acetylcholine, and this is stored back in the synaptic vesicles for the next time. So, to sum up, there are two systems which control bodily processes: the nervous system and the hormonal system. Chemical mediators affect cells in their immediate vicinity. Plant growth factors are like hormones, but are made throughout the plant and can target all cells, not just faraway targets. IAA is one of these hormones and is responsible for the way a plant will grow towards the light. Neurons pass impulses around the body and are classified by how they function. Resting potential is when the inside of an axon is more negative than the outside due to the active transport of sodium ions. Action potential is the impulse that is passed along the axon. Unmyelinated axons pass the potential slower than myelinated axons due to a lack of insulation. The refractory period between potentials stops impulses being sent continuously. The synapse is the junction between neurons and effectors. Acetylcholine is a neurotransmitter that causes the action potential to be regenerated. Thanks for watching. Science with Verity.